All right, let's get going here. Welcome to the third and final workshop on DNA analysis that we're doing here this semester. I'm really excited to kind of go over, kind of bring everything, tie everything together here and show you some cool new stuff at the end. So we've kind of had a journey where we started out in the first workshop where we were, loading mutation data and trying to understand the different formats and, trying to get some really quick visualizations like oncoplots, lollipops. And in the second workshop, we went on to more looking into data a little deeper, right? Doing some statistical testing, clinical associations, survival analysis. So now we're going to kind of bring this together and we're going to tackle just some fascinating questions that often face us in cancer genomics. And these questions are, now that we know kind of what's going on with mutations, what biological processes are actually caused by these mutations? And I also want to look today in the sense of tumor evolution and try to understand through phylogenetics, clonality, mutations, how did this tumor actually evolve in the first place? And also, finally, based on our mutation patterns, can we actually predict different cancer types? So I want to just kind of introduce you to some different ways that you can do prediction models to kind of get you thinking about how you could apply that to your work. So, we talked a little bit about mutations before. As far as the process, what a mutation is, obviously. And, we think of them as we talked about as, every tumor, as everything it goes through over time, and it's just being barraged by life, environment, food, differences. The tumor goes through these different time periods, and obviously it carries the scars of the biological processes that shaped it. So now we're going to want to look into that history of the tumor to try to understand more of what's going on in the cancer and possible treatments, right? So just think about big picture tumor evolution, just in general here, to set the stage for the kind of lesson. So, we start off with a normal cell, it's just kind of cruising along, doing its thing. It has some germline, like some pasture mutations that is collected over a lifetime, but nothing's really happened to it to cause problems. Then it kind of builds off and has some early mutations, very early clonal mutations. And clonal by the sense that the mutations are found in a high number of the different, parts of the tumor. Because the mutation happens pretty much across the entire tumor. You have very high allele frequencies, things like that. And things like, we'll talk about colorectal cancer, you have this early APC, KRAS mutations, these initial drivers to push it along. And then as we move along and we evolve, we end up having more subclones. So that's just more mutations in smaller parts of the tumor. As the tumor evolves, it's going to have separate mutations than from the bigger herd of all the clones, right? So this kind of develops these subclonal branches in the tumor tree, right? And then as this kind of builds more and more, we end up with this very heterogeneous tumor mass. And as you all know, that's the problem with cancer, is it's very heterogeneous, right? If there was just one mutation and that was it, in the whole thing, it would be pretty easy to treat, right? But that heterogeneity is what causes a lot of the difficulties and things like treatment. So what will we cover today? Well, we're going to look more into mutational signatures. We kind of touched on the topic at last workshop. We're going to dive more into this, and look at really what processes are actually caused by these mutations. And we're going to use the colorectal TCGA bulk cancer data set for that. And we'll also want to pull out our own signatures, right? Can we extract new, novel signatures using MFF signature extraction? And what mutations are clonal and what are subclonal? So let's just look at the clonality analysis to start diving into the evolution. And then we'll step further into tumor phylogenetics to see how clones evolved. And then we'll go back and kind of build upon last time's driver identifications to see what's driving. And then we'll finally go into the prediction like we talked about to see what we can actually predict. So for our 3 data sets today, we're going to be using number 1, primarily the TCGA COAD data set, which is a really good data set to work with. It has a lot of different interesting features. It has some micro instability, these MSI tumors, which have mismatch repair. You're going to see things like SBS6 and SBS15 signatures. We have very high permutators, the pole mutated mutators, where the pole associated enzymes can cause a lot of early very high mutation burden, and they end up having SPS around 10 usually. And then we also have copy number instability. We often see in really all tumors, but we see it in COAD a lot. We'll see SPS 1 and 5, things like that. And really, we're the cool thing about COAD is very well known, the process of mutation. So APC happens first, then KRAS, then TP53, then the others are in a more some clonal nature. So these three guys will be the clonal drivers of the tumor. The second data set, we're going to jump into an AML data set. We want to look at phylogenetic trees, and there's a really simple data set to work with to understand the very basics of this. We're already jumping into single cell for this. Like I mentioned, this Mission Bio Tapestry AML data set, one of their first data sets, and we will be pulling off this paper as Miles et al. Nature Genetics, and we're actually going to be doing a synthetic data set to give you a cleaner lesson today. And third, we're going to do pan-cancer mutation data. So we're going to pull in, I'm going to use this new package to pull in MAF files easier, and then we're going to look at 5 different cancer TCGA data types, and we're going to look at the mutations and see if we can actually predict cancer types from these mutational patterns. So that'll be kind of cool. So we kind of talked about the learning objectives here. So let's just jump right in. First thing we want to do is set up our environment, right? Our analysis environment. A lot of the packages are the ones you've seen before. Best thing to do is just jump in and load, and the ones you don't have, then load after. You'll be having a few things from BioManager. You'll use BioManager install, and then obviously the CRAN install. One other thing that we've done before, but I wanted to remind you, whenever a package is not on CRAN or Bioconductor, but it's being developed, like usually on on a GitHub website, there's a package called remotes. And if you install that remotes, it allows you this command, remotes install GitHub, and the GitHub page will have the directory to look at, and that's how you can install packages if you didn't already know from GitHub itself, and that's one of the packages we're using. A lot of times you wanted to do this to get early developing packages that are still in development, or ones that have new features that haven't reached your package as well yet. So then after that, we're just going to load all our packages, give you a chance to kind of look over later, see what's really going on with those packages, but you will see them used here shortly. And first, we're going to jump into our COAD dataset, our TCGA dataset, right? If you remember how to do that, we're doing exactly the same. I'm copying the code exactly except I'm changing the word COAD here from PAAD or BRAF or BSLA or whatever. And you can use the same code here, and we're just basically we're querying, downloading, and then formatting the MAF file on something we can actually use. And we end up with the COAD MAF file. If you want to look at that, if you remember what these things look like, there's the COAD MAF there, and then we have our data, right, in our data column, where we have all our genes and our positions, our type of mutations, things like that. We don't have the clinical data here today, but we could pull in the clinical data as well. But if you remember, that's how we kind of look. We really pull that data column, our data list is what we really pull it used for. And we have 1,000 samples around, and we have quite a few mutations to work with. So that's it for that. For our Mission Bio data set, remember what Mission Bio does is a high throughput mutation sequencing. It uses a targeted single cell DNA genotyping with amplicon-based sequencing. These are really targeted panels they use. The thing we're going to do today has 30 different genes, so we're going to look at 200 different cells. And this all comes from the Miles et al. Nature Genetics paper, and we are going to simulate that data to make it a little bit cleaner and easy. So we have the same genes, the gene panel, and we're going to set up basically a representation of how those clones will be set up based on the paper. But what you think of? Just think of single cells. We have 200 single cells, and across that we have 30 amplicons that we're going to be sequencing. And amplicon sequencing, thinking of it as like targeted exome sequencing. We're basically putting probes in around those genes, and we're only sequencing the areas of those specific genes. So it's a very small panel, right? So we can get a lot of reads and a super, super high depth to feel really good about this. And that's for things like trying to find things like FLT3s, ITDs, things like that. You really want a high depth for this. And we can move on, also Mission Bio moves on to higher thousands and thousands and thousands of cells for some of their cleaner new operations. But we wanted to keep it kind of simple for us here. So we basically I kind of just simulated the data. Don't worry too much about that. And we have 30 amplicons and 200 cells, 26 normal cells, and then we have 3 clones here that have different kind of representation distributions of genes that represent that paper. All right. So, and then we'll pull in we're using the bladder cancer data earlier, and we will talk a little bit more about that. We're going to be pulling that in here. We're pulling in the pan cancer data set soon, and then we'll pull in the bladder cancer actually for the practice problems, and we'll talk a little bit more about that as well. But for now, let's dive into our co-ed data set and start looking at mutation signatures. So last week, we briefly, the last 2 weeks, I guess, we briefly talked about mutation signatures, what it means for you to be a purine versus pyrimidine versus a C to T versus, C to G, different kind of switches between the A, T, C, G. And we kind of saw how that could have an effect on the cancer a little bit, but we didn't really dive in to really talk about what is going on. And we want to do that today. So, right, think about every cell faces constant DNA damage, right? From all sorts of different things, from endogenous sources to exogenous sources, and it leaves a kind of a characteristic fingerprint on each cell called a mutation signature, right? And this could be as opposed to looking at just one mutation, you're looking at all the mutations, whether they're synonymous or non-synonymous. And that kind of basically builds upon that mutation signature. And we are doing SBS, single base pair signatures. And here's a few, just the common ones you will see a lot. SBN 1 and 5 are very age-related signatures, and you can see these. If you look at these, you'll see C to T or C to A. That's the transition from the base pair, right? And then it gives you the site you normally found. Like CpG, the P is the phosphate, so you have C, phosphate, G sites. You see a repetition of those. That's where you often see these C to T changes for SB1. That's kind of how you look at this stuff. But just to jump in overview, like 213, ApoBec signaling, problems with that, cell cycle issues. SVF4 tobacco smoking, MMR, mismatch repair deficiency that creates microsatellite instability that happens at 615, 20, 615 most common, UV light, 7, and the POL enzyme issues that lead to proofreading deficiencies and cell cycle issues that cause hypermutators. Both MMR and POL cause hypermutators, and then you see SBS 10 usually for POL. Now, this is really important. So last time we talked about how do we decompose all of our different signatures in a way that are kind of simple. So we found if we just go from C to whatever or T to whatever of our ACTG, we can redefine all of our different guys in all of our different transitions in 6 different categories to look at. Now that's powerful, and that's kind of what we looked at last time, and we saw some different things in there, but the flanking bases actually matter important too. Mutations don't happen in isolation. So what we also want to do is look at the flanking, the 5' and 3' bases for each of the actual signatures, right? Now, if you remember, you have A, T, C, G, you have 4 different choices for each one. So that's 4 times 4 is 16, and then times our original 6. So 6 times 16 is 96. So really we have 96 different contexts, trinucleotide contexts, to be able to classify a certain cell, right? Or a certain group of cells or samples. So remember, we have 16 different contexts from each of our different, transition types. Now, okay, so how can we extract that out? The full 96 thing out, instead of just a simple one? Well, here's what we can do it. Math tools, right? That was a simple way we did things before, so let's use that again. So there's a function called trinucleotide matrix that you can just put in your math file, put in your reference genome, right? And usenth is use synonymous mutations. You want that to be true because we want synonymous mutations. We want as much as possible. And we see the kind of the output here, it gets going. And it already mentioned some APAC stuff, so we want to probably look into that as well too. And we get our matrix that's our all of our samples and then our 96 different contexts, right? And we'll just plot those out. And here's what you're going to see is a really good plot here, right? So this shows all of our different 6 changes, and you look at each 6 changes with all our different ways it could go, right? So we have our 16 different contexts, all our different flanking situations, right? So we have our 6 different major situations, then our 16 different flankings for each one of them, right? And then we see kind of a super, it's like superimposed upon each other, all the different signatures, right? So all the signatures right now are found. Like for instance, you have SB1 around there. Everything is found in here. But that's the problem, right? Let's say there's 4 signatures. Well, this is just like the addition together of 4 signatures. How do we decompose this to actually find the 4 signatures that when you add those back together, you see this? So that's the goal, right? So the first problem is like, how many signatures do you pull out? Because the number is extremely important. Think about it, because it's the decomposition, right? So if you choose too low a number, you're going to miss out on a lot of the signals. Too high a number, you're going to be splitting signals. So that's something very important. So we're going to try to decompose this down to smaller areas, right? And remember the APAC stuff? One cool thing about MapTools has a lot of stuff like this: plot apobac difference. So you put in the math file and your 2TM, your extracted trinucleotide matrix, and it'll jump out and tell you a lot about what's going on in those for apobac. And there's different functions too. But what I wanted to show you here is we have flags right away. Why would these be flags? And we'll see that in a second. The reason these are flags is because they're hypermutators, and hypermutators throw this whole analysis off. So we look at here, we're seeing 366 to 670. We're seeing it. I mean, this is like, 1/3 of all the samples are apobac high, which is not the case in CoAD. It's much more like 5%. So we're seeing here that these hypermutators, since they're mutating a lot in general, their baseline's higher, right? So they're making it look like there's more mutations that don't really relate. Of course there's mutations there, because there's mutations everywhere in that sample, right? So then you can't count those as apobac. You gotta pull those out. So that's a really good, important thing to see, is we'll often see these hypermutators mess up a lot of our analysis. So we know that this is probably a decent amount off, and then that's good to know starting out, right? You can read a little bit more about it here, what we're talking about there. All right, so now we have our signature, right? Our trinucleotide, superimpose additive signature, right? So let's try to extract out all of our real signatures out from that big number, right? So we'll keep using MacTools, and we use the extract signatures function. Put our TNM matrix one again, our trinucleotide matrix, and we're just going to guess for now. I'll show you how to make the number, but I want to see what happens if we just guess 4. So just 4, we're going to assume there's 4 signatures. We get our sig results, and then we actually just plot the sig results right here, plot signatures, and we'll get all of our signatures that we see. And if it has, we'll look at it's just basically taking that signature and seeing how similar it is to all the cosmic signatures. And it's giving us a cosine similarity value that if it's above 0.85, it's a pretty strong match. So when we look in here, you see here, getting blind here, we see 2, 5, I believe, right? 6 and 10. 2, 5, 6, and 10. So 2, if you remember, that's apobac related, right? 5 is kind of that aging you see a lot. 6 is the MMR, MMR DNA mismatch repair. And then we have 10, which is the pole enzymes, right? So we're hitting exactly the kind of things we would expect to see in colorectal cancer. So we already feel pretty good about those. The question is there really 4 or not, right? So, so I talked about, we have SB1, it's aging, SB615, the fact that we have 6, that is our MMR, and then 10 is our pole. And so I feel really good about those. So what that did is it took a de novo signature using NMF and then compared back to Cosmic. So what we want to do is MapTools is great, but we want to use kind of a newer method, the kind of method we mentioned last workshop, the SigMiner, which is kind of a more gold standard. And it gives you a nice, cool plot. There's a lot of things you can do with it. So we're going to do the same analysis with SigMiner, a little bit of a tweak to show you what we can do. So, the Nova way, the NMF way, like we use the MapTools, is better for discovery. Things like SigMiner, what it's going to do, it's not going to do that match. It's just going to distill, pull apart our signatures, right? It's basically directly decomposing each sample, like unmixing paint into different colors. So it happens at once more than this 2-part de novo and then comparison. Anyway, so let's get this going. So the first thing we do is just like last week, we do a signature tally, put our math file in, and put our reference genome and our synonymous equals true. So very similar to last time, except we added this. And then we have our mutation tally of our signatures. So we have our tally just like before, right? All our samples, and then our 96 for each one, or for each signature, we have our representation there, right? And then, so we basically want to grab that, and we're just grabbing the list of all the matrices, and then by each matrix, we want the list of their 96 guys, right? So basically, we're finding all of our matrices, We went to the SBS 96 for each one of the samples, right? You could also put a $sign MF matrix here as well too. But you can just pull this line right there. It'll always be the same. We're transposing it, and this is our input matrix. That puts us into our sigfit function that actually finds the signatures. It unmasks those signatures, right? So we put that in. We're looking at all signatures and just our latest, our GR chromosome 38. And basically that gives us our fit. We could go with that fit, but we want to kind of filter this out a little bit. So what we're going to do is sum the exposures for each row. So we're basically looking at pulling all the signatures across all the samples into one column. And then we're basically getting stricter, and we're basically filtering out here. We just kind of sum the exposure, and then, excuse me, we sum the exposure. So we want to know how many mutations are involved, and we're basically taking just total exposure that has more than 5% of its mutations that's toward the signature. So we only want to look at signatures that have over 5% of mutations going toward everything, right? Because if there's just like one mutation going toward it, it's pretty unlikely for that to be anything, right? So we're just kind of setting a standard of 5%. So here, and then we're basically calling that active signature, and then we do show sig fit. We call our fit, and then we add an extra thing here because we wanted to just grab the active signatures. We could have just put fit alone, it would give us everything, but we want to filter down. We could also filter down here too to put in like n equals 4, 5, 6, 2. We can do that as well too. I just wanted to show you a cool thing that we could do by throwing a wider net. So we were very strict with our first one, right? The NMF, extremely strict. But I wanted to throw like a wider net this time and really make sure that I was getting, not missing anything, right? So that's why we wanted to see, to filter signatures that are contributing over 5% of total mutations, to look at really biologically meaningful signatures. So here's our result. What do we got here? So first, look on the bottom here, we see 1, 2, 6, and 10. Well, 1 is the same, very similar to 5, so it's just like last time. And then we have 2, 6, and 10, just like last time, right? So we're basically getting the same signatures last time. However, since we've not made it so strict this time, kind of opened it up a little bit, we found extra signatures. Now, the danger of this is if you throw too wide of net, you could start finding signatures that aren't real. If you throw too small a net, you're too strict, you might lose out and miss out on real signals. So you kind of need to look at both and get a feel for what makes sense biologically and statistically. So what do we see here that jumps out at me? We see 10A and 10B. So that pole signature is being split into two now. It's not just saying 10, it's saying 10A, 10B, right? And it's saying 10B is actually stronger. So we're getting more fine information about the distribution, the ratio of pole enzymes, right? Now 2 is apobac, right? But also 13 is. So now we're getting a little bit more refined idea of what's going on with the APAC signature. And then MMR2, the MSI stuff, 6 and 15, same thing. So really, this is really an awesome result, I think, because if we tighten it up, we just get 2, 1, 5, 6, and 10, but we miss out on some of these other signatures. So by widening the net and loosening it up a little bit, I think we've really found some interesting biology here that makes a lot of sense. So I think that's really cool, like here we're seeing our 1 and 5, we're seeing our 6, we're adding our 15, we're adding our 10A, 10B. I think that's really, really cool. I think we're really finding some cool, real stuff, right? So it's a good thing to know, right? Now so we'll be talking a lot about NMF and these two methods. What does that even mean, right? De novo signature extraction with MF. What does that really mean? So this is exactly what we did with the MAP tools. And it's exactly what SigMinder does behind the scenes, but it's a little black boxed. So I wanted to kind of explain to you what's going on and take you through the functions of the SigMinder kind of behind the scenes to see where we're getting, right? Sound good? So what the heck does NF mean? NMF mean? You hear that a lot nowadays. It's not just using mutation signatures, it's used in a lot of different biological clustering, and it is non-negative matrix factorization. Once again, what does that even mean, right? So basically what it's doing is it's decomposing a matrix into 2 smaller matrices with all non-negative value. So if you multiply those matrices together with matrix multiplication, you should equal the original, right? So what you're splitting it up into 3 different matrices. One's just your samples times your context. That's the matrix we've seen, the 146 times 96. Then we're going to have an output matrix. This is going to be samples and signatures. It's going to give you more of your contribution of each signature. And then your H matrix is really going to be your actual profiles. So let's think about this in plain language, a way that I think is really cool. Let's say you're listening to an orchestra playing, right? But you want to figure out which instruments are actually being played and how loud each one is. What's the contribution of each one to the sound? Well, this is exactly what NMF does with this mutation data. It takes us this complex mixture, the orchestra, right? And it decomposes it into the signatures, which are the actual instruments, and then the volumes, which is actually the exposures, right? So that's why you see this ACE matrix up here is really the signatures, and then our the W matrix is actually our exposure, our contributions, right? So W tells us what the H tells us what signature profiles exist, and W tells us how much they are contributing to each sample, right? But what does non-negative mean? Well, so other decomposition methods, let's say like PCA, they allow for negative values, but this is good in some contexts, but here in this biological additive context, this would mean you have signatures that are harder to interpret. Like you have to have a negative C to T mutation, something like that. This is, in clustering and some RNA stuff, it's a little bit easier to make sense of this. And you could do it with the normal way. It's just harder to interpret. And the whole idea of the signature analysis is to make it easy to interpret it. So by restraining the mutations to positive values, you're really looking at more of a real world situation, right? Where each signature is an additive component, just like how the total mutation spectrum of the entire tumor is the sum of its contributions. Well, each signature is one of those additive components. That's how we're able to break this down and look at it, right? And it gives us a much better understanding of what is going on, and also allows us to look more into novel values of the signatures. So we split these up into our three matrices, just like I mentioned. First thing we're going to do is just get an estimate of how many signatures. Let's actually statistically decide how many signatures to choose and not just guess, right? So we're doing a sig estimate just like before, and we put it in our matrix, but this time we're doing a range, 2 to 8. So we want to do 2, 4, 6, 8, right? And we're going to look at a few different representations. You see here, you'll see dispersion, complement, you'll see different things here. And this, so the diagonal is all the different metrics we did, and then here are all the different results here. I know it's impossible to see, but just wanted to give you an overlook of what's going on. The four things we really care about are this comphonetic, dispersion, silhouette, RSS. The first three, you want the highest value, and you usually see a strong dip after that highest value. RSS, you want more of an elbow. You can already see here, if you look at 3, it looks really good. You see it goes higher, higher, lower, higher, 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 lower. So we're really seeing a drop off in the first 3 after 4, just eyeballing this. It's a little bit harder to see the RS, so let's plot these and see what they look like, right? So the cophonetic one, high, so this is 2, 3, 4, 2, 3, 4, 2, 3, 4. So all 3 of these look good. RSS, it could be 3 or 4. I mean, you have to kind of think of when that elbow happens, this is a tough choice. I want to say it's more like 3. I can see how they're saying like they're predicting 4 would be there, and it kind of smooths it out. But I think 3 is a value here that's not too crazy either. And when I first did this, 3 was showing up earlier with the map tool, so it could be 3 or 4 on this deal. But since everybody else is 4, we're gonna go with 4. It looks pretty good, right? And read more a little bit about what these things are. These are all things we've looked at before as well, especially like silhouette and RSS. All right, so now let's actually take out the signature. So if you saw here, we did a sig estimate, right? To get this, right? Sig estimate, and we want to actually pull the fit out. So sig extract, just like before. We have our matrix. See how I did an NF matrix this time? And instead of making an input matrix, so I have sig extract, optimal K. We've decided that to be 4. We can just put a 4 there. And it's going to run through, and now it's going to have a little bit more restraint there, And now it's going to go through and find 4 signatures and split them apart for us. So 1, 2, 3, 4, and it's going to get us, what we're going to do is compare these, get signature similarity. We're basically going to compare this de novo signature to cosmic signatures to see how similar they are, right? So we kind of do that here. And if you look here, this is all of our best matches here. And remember, you can always just copy code like this. You don't have to write everything, just copy a lot of my code and as you build your projects. Just like going to Stack Overflow and doing it. It's the same thing. There's no harm in it. So let's see what these match. So signature 1, you see it matches these guys here by similarities. And we see the SB, we see right away, we see 2, 5, 6, and 10, which is exactly what we got the first time, which makes sense because we did it pretty much the same way. So when we restrained this guy, we got the same result for MapTools and SigMiner, which is very restrained. But remember what happened was, right, if you go a little bit closer here, you see that 213 is not off, it's just 0.62. So that's why we had 213. When we had that the 10A, 10B, they're very close too. And then, you see 6 and 15 are really close. And then you see, 1's pretty high here with 5. One thing we lost is the actual 5 itself. But rather than that, 5 itself right there. I'm crazy. So everything matched up to exactly what we thought. And you also can see how if you can look a little below and see the ones that with a wider net might be biologically important, important too. Because things like 0.85 for 15 is really still really high. So that's really going to be an MMR result you want to look at. So that kind of gives us what we saw before, this distillation until these curated signatures. But here's the coolest part. You can make your own signature, right? So I'm just looking at a few things here. You can make this even bigger for signatures, but I have basically my own 4 signatures that are basically made up of additive values of correlated cosmic signatures, right? So signature 1, for example, has 1, 6, it's really strong, and 6, 15, and 1. So that's aging, and this is MMR. So signature 1 is very MMR related. Signature 2 is very pole related. Signature 3 is very APOBAC related. So you really see how these signatures are kind of pulling apart, where the other guy is more just general, signature 4 is kind of more general colorectal cancer. It pulls in things like 40 for the, I believe 40 is still MMR, right? It pulls in a lot of different things, right? But what's cool is you can kind of pull out these signatures. We can combine these signatures into one. We can take parts of them. We can look a little farther, do some more analysis, and come up with our own signature. Like, for example, when I was working in lung cancer, we were looking at particulates and pollution. So we wanted to see, is there a mutation signature among parts of the tissue that have pollution versus parts that don't, right? So we separated those out and compared them and found that there was a unique de novo signature within that polluted area in the lung. So it's just kind of an example of how you could go look for your own signature. That's going to be a very good thing to raise your paper up into a higher paper tier. All right. So, and you can also look at how this looks over all your samples. So now we're just looking at the distribution. So all these samples have a high signature 1. These have a high signature 3. We could sort these more by signatures, and you could get a feel about what's the distribution of these different changes in your samples and look farther into what other things could be going on in those samples. DNA, RNA, copy number, different things. But just keep in mind that a lot of this is going to become, these hypermutators are leading to this. All right. So now we've kind of moved into our signatures, things are going on. We're kind of giving an idea in the back of our mind about what the clonality can be. Let's jump a little bit farther and start looking at this tumor evolution. And we'll start by looking at clonality and VAF analysis, which is just variant allele frequency. And if you don't already understand this, just think about, let's just think about a T53 gene, and we're looking at one base pair on that gene. And so we did exome sequencing on that area. We have 100 reads over that one base pair, right? If half those reads are mutant reads, so what you can imagine a DNA, you have one strand that's normal, the other strand's normal, except that has one mutation on it, right? If we looked at that exact spot, we would get half normal reads and half mutant reads, right? On average, because that's what's being expressed. just like so that's giving you your kind of your variant allele frequency there, right? 50%. If they're all mutant, then every single one would be done. That would be homozygous, right? That would be all your variants. If only 1 out of 100, you'd have a 1% VAF, right? Which would be a very small, very small subclonal mutation that could be useful or not, right? So that's kind of how you think about these VAFs. So once again, if you have about a 50%, that means you're heterozygous, right? Half your alleles are mutated at that spot, right? A lot of those can be, really early drive mutations. If you could have 100%, you can get dual knockouts that are homozygous, or you could have an LOH and one mutation, which is kind of the same thing. You've lost everything on that gene. It's all variant alleles, right? You can go and really clonality is complicated. We're dealing more with homozygous, heterozygous, right? But I also would say clonality is probably 40% higher. 40% to 100%, I'm thinking of things being really clonal. It's kind of subjective. 20 to 40%, definitely subclonal, a smaller, late rising mutation. And then, very small, low frequency subclones, like under 20%. This could be like a very, very late mutation, or it could be, therapy resistance, something reacting to, a treatment. So that's kind of overview. If you have different complicated copy number events, things get a lot more complicated, and we'll talk about that next time in our next workshops with copy number. All right, so let's just look at the VAFs and TGJ coads. So I don't worry too much about this, but I just kind of looked at the data. I kind of calculated it myself. There's a way you can just pull it out by itself, but I basically just took the alt counts and divided them by the ref counts plus alt counts, so pretty simple. And I got my mutation here. So what's interesting, what pops out right away is, why is the peak at 0.25? Now, it's a really good question, and this is about tumor purity, which is something you have to go into when you're dealing with the VAFs, and it becomes very complicated, and we'll kind of leave that for another day. But if a tumor is 100% pure, you would expect this curve to be around 0.5, right? The reason we're getting 0.25 is because this is not pure, right? It's really saying that it's about 50, but this is implying 50% purity. This is kind of complicated because it's not one patient, right? This is one patient, and we had a different regions, or we had single cells. We would be much closer to 50, I guarantee. Since we're putting a lot of patients together, it's throwing this off a little bit. So just think of this as one patient, or just even the group itself as a conglomerate. It's showing a lower tumor purity in just the samples we happen to sequence, I guess. But however, the theory is still intact here. If you have, over 50%, it's going to be over here. It's going to be clonal. The guys down here in the very right, this tail is long tail, where we have a lot of super clonal mutations at the end that happen really early on. And they also have a double hit, so they're really driving the cancer. And these guys, we have, early on clones as well too, but probably more single hits. As we move to the left here, we're looking in that 20 to 40% range, and we're getting the subclonal mutations. And we see this drops off really hard. There's not as much of a tail here. And part of this is because we have to hit a 0, but it's just not as much of a distribution of these kind of mutations as there are of the clonal ones, right? So we have 20, 40%, under 20% is kind of that low frequency mutation. So that's kind of how you can look at this. And you can think of clonal mutations, they come first in evolution, and then subclonal ones, and then frequency. So you can kind of think of this right to left is kind of how the actual tumor is evolving at a high level scale. It totally is a good representation of that. So it's always good to plot your EVAFs to see if anything looks off, get a good overview of your tumor purity and your evolution. All right, and we see here that if we split up a little bit more, the subclonals until minor and major, which is anything over 20% here, we see that the majority of things are put into clonal and major subclonals, which makes sense because you have these beginning mutations, and then as you move on, you end up having late emergent mutations and probably therapy resistance, things like that. So this makes total sense as far as a biological perspective. And also the key genes. Look at the VAFs, the key genes. I didn't look at the significance of these, but just if you kind of look at this kind of a hand wavy, we're seeing TVD3, KRAS, ABC, all of being the highest, right, among these genes. And we're seeing, the VAF ABC being around, it's getting there, 25 divided by 2, 12, 32. So, the median's 32, but, there's plenty up here that are a lot higher. Same with these guys. There's just a big heterogeneous distribution of these mutations. But the guys up here are probably found, these are very clonal mutations, right? So we're still seeing clonal mutations, high frequencies that lead to these, tumor suppressors in our oncogene here. Oncogenes, back and forth. Those things make sense to be driving our cancer. If you kind of look at just a simple overview, once again, in clonality, we're seeing in the left guys, the far left ones, we're seeing distribution that are very clonal and really don't have that much clonal stuff here. Toward the end, much more clonal with less subclonal. So it just kind of goes back and forth. In the end, it's only subclonal. So it's very, very small amount of the clonal mutations left in there. They definitely are left in there because they have to be, right? Unless there's a back mutation. And this is just showing you kind of an idea of your inner tumor heterogeneity as well in your different samples. So most samples are more heterogeneous than others, and they can be treated different, right? Okay, so let's take this. So one thing I want to talk about is so far we've been dealing with bulk DNA. And you may wonder, why have we not done single cell? Well, working with DNA is a lot easier than RNA in the sense that you can treat, whenever you call in your mutations for one sample, you go through a lot of pre-processing and a lot of post-processing, and you get a good feel for the mutations of that sample, right? Batch effects don't bother you as much in this sense. So if you have multiple samples that have happened at different places, and the mutations have been rigorously called in those mutations, it's a lot easier to put those samples together, cluster them and work with them, and treat them as single cells. So you can almost think of, if you have a bunch of bulk cells put together, bunch of bulk samples put together, that you can treat those as single cells in general, right? It's not that easy, but just in general. Now, in RNA, you cannot do that. You cannot grab a bunch of bulk RNA data and stick it all together and treat it as single cells. It's completely different the way you normalize and handle that data. It's a lot more complicated to deal with RNA in a lot of ways, with batch effects and different platforms and different things. So that's much more complicated. So really, so far, since we have been dealing with bulk, but really you could have treated it all as single cell data, and the same concepts can be applied to single cell data. Is my point, by just treating the samples as single cells. Okay, long story there, but when I want to show you phylogenetics, bulk phylogenetics is extremely difficult. And while it is fruitful, multi-regional sequencing, things like that are very interesting. It's much harder to build those trees, and it would take we'll have to make our own full phylogenetic workshop to talk about that, which I really plan on doing later. So I really want to jump into using single cell data and phylogenetic data at the hopes that you'll be moving toward that, excuse me, and it's much easier to use and makes more sense, because you can't put a bunch of samples together and say they're evolving because they're not the same sample, right? It's very hard to do that. Where one sample, all the cells are in the same evolutionary pressure, it's much easier to understand what is happening, right? Now, that being said, I've built trees and done a lot of work with bulk multi-regional sequencing, and it definitely is possible. It's just above our pay grade for today to talk about. Anyway, so let's talk about our single cells, our single cell information. So what we've done is we just made a binary matrix. Let's see our single cell matrix. I'll show you in a second what that looks like. This single cell genotype matrix. I want to definitely show you what that looks like. So this is there's two ways you can do this. You can go by VAF, or you can go by a binary. So I'm going by binary. So we have a 30 gene targeted panel for the Mission Bio, and we have 200 cells down here. And what we're doing is a binary. We're saying for cell 1, IDH2 is not present in any of these genes. And then we're saying for cell 3, a 1 means that it is present. Now, we've totally simplified this. And as you can see, that we have plenty of 1s, right? But we don't have any 2s. So another way to do this to make it a little more complicated is you could have said 1 for heterogeneous, 2 for homozygous, 0 for normal. We're keeping it simple today, and we're just saying if you have the mutation, it's 1. If not, it's 0. This is a very common way to make a sparse matrix to handle DNA data. So what we've done is we started here, and we're going to do a quick TSNA. So whenever we have mutation data, single cell, I like to do a TSNA plot, see what we got, see if it clusters well. So I'm basically just doing our just like last semester, doing PR comp. I'm just basically scaling our data and doing our PCA. Took the first 20 components, And then I made basically I ran a TCNA on it, very simple, and then I went ahead and plotted that. And what we got here is a really good result. As to be expected, this data is really, really clean and has very few mutations that are very powerful in separating clones. We see our normal cells here, and then we see kind of our other ones split off. Now, remember, this is TC&E, that you can't really say like, oh, these are closer together, we should use these, or the others should not. And I like using TC&E for smaller data sets and map for bigger ones, but you can go either one. They're both equally as good. But I think this does a really good job of separating our data sets as it is. So let's go a little bit more, and now we're going to do a hierarchical clustering. So the first thing I like to do when I get data is look at the clustering and then the hierarchical clustering. Right? The clustering, the visualization of the clonal architecture, and then actual cluster the data with our clustering. So we just took our cell metadata, I arranged it by clone and just by cell ID. And so what I want to do is I want to keep our clones. I want to make a matrix, and I want to sort. I want to build a tree that sorts the top and clusters the top. But I want to keep our clusters we already have for the left side. So that's why I'm arranging them by clone. And then I'm basically just pulling a cell ID and basically just plotting by that, doing a feat map with my matrix by that order. So I'm setting the cell order because I want to keep those clusters, and then I'm just running feat map. And I am, I have my annotation rows have a certain color, and I am clustering rows equals false, right? I want to keep these the same. Clustering columns equals true. Okay, so these are all clustered, these aren't, but we have our clusters, right? And that's what we expect, right? 3 clones, that's what our data has told us it would be like. So let's kind of check it out. So we have our normal cells here, no mutations. So that makes sense. If you see little tiny ones, that's just dropout or noise that we've simulated. This to be expected in data sets like this. Second thing I see, 1 mutation is found in every single one of these. And it's, what is it, DNMT3A, which is a very common early mutation that's found in AML. So this is what we expect. We found it in every malignant clone, so we can say this is the founding mutation. After the normal, this was the founding mutation. It exists in all the clones, right? So what happens next? Well, this guy seems very separated. So what's going on with these guys? Well, there's one more mutation that they share. This, what is it, NPM1? NPM1, right? They both share this. So that tells me that the normal cluster is first, right? And then that these clusters are more similar to each other than this cluster because they have another mutation that this guy doesn't have. So this guy's split off early on. These guys are more together. And we will go ahead and see that when we plot it out. So let's actually make the tree. Let's make a neighbor joining tree and see if this actually runs out. So if you remember how to do that, the first thing we got to do is do a distance. So we just have our matrix object that I just showed you, right? We're going to use the Jacquard or binary, the best one for binary data. And we'll get our distance. And then we'll do NJ for our tree. Now we have our tree, right? Now we could plot that, but we actually know the root, right? So let's root the tree in the normal. We know the normal. We have a normal. So let's root this. It makes it a lot better tree, right? So we do cell metadata. It's just all our data, right? And we just took out the normal. So I want to know the IDs of all the normals, and then I'm just going to use the very, very first one because I already know some of these normals have error in, some error in them. So if we grab the entire normal population, it would throw it off. So we need to take cells we know have no mutations. And I happen to know the first one doesn't. So all I'm doing is we've made our tree, distance matrix, neighbor joining tree. I just grabbed the first cell. And I'm basically just rooting the tree. After that, I root our neighbor joining tree. And outgroup equals the normal cell, the very first cell. And I'm just telling it to resolve the root. And then I'm going to laderize it next, like I mentioned, just to make it pretty. And here we go. Here's our result. Here's our normals that we rooted in. And here's our 3 clusters, if you want. And we're just going to, color these things, take a look at them, and get a feel for it. So remember, this is neighbor joining tree. So neighbor joining, what it does is It really goes through all the different ways you could make splits, and it makes a tree that minimizes the branch length for the whole tree. So it doesn't have a molecular clock. It allows evolution at different paces, but it's not looking at how many mutations or evolutionary processes are making any assumptions. It's just trying to find the tree that it has the minimal overall branch length. So tree looks good. Here's our 3 different clusters. Now you see some gray in here, So these are some of those clusters, some of those cells that have been misclassified because they have some noise in them. And that's to be expected, and they're just outliers, and they can be thrown out or kept in either way. And then we're seeing 3 major clones, and what we're seeing here, I think this blue clone is the first one, right? That's separated. Okay. So what happened here? These are kind of split off normal. These are split off normal here. So really, what's happening is there's some error here of these branches, but really the main split is happening here, where the blue's going that way and green and red's going that way. So what's happening here is that MP1 mutation is probably happening, and that's separating them. So this guy goes on without that mutation. These guys go on with it, and then he ends up having some more mutations. And then these guys both have the MP1, so they're stuck there. That's an outlier right there for the gray cell, right? So they're back to here, right? And then this guy has its own mutations. Really, it goes FLT3 ITD, FLT3 KTD. So that's that last split there. That's kind of how we can kind of look. We can kind of follow our way through it and kind of get an idea of what's going on in the tree. Now, since we've done that, let's try some more trees. Some things may work better than others. The next thing I want to try is the maximum parsimony tree. Now, what this looks like is it looks over the whole tree, and it looks at it more logically in a molecular evolutionary way, and it tries to find the tree that has the least possible mutations, because the simplest is normally the best. To do that, I use my single cell genotype matrix, just like before. I make this what is called a phi-dat matrix. You have to use this for some of the more complicated trees. It's very simple. You just use user levels 0 and 1, because the binary is 0 and 1, right? And it basically makes my file, and then I use this function called Pratchett to make the tree, and I put that in there, and then my tree's done. Then I just root it again, just like before, and I ladderized to make it pretty, and I plot it. And here's where we go. And now the maximum parsimony is a unimetric tree, so everything goes down to the end. Everything is the same length. The tips are all at the end, and it's basically chosen the fewest total mutations using Occam's razor kind of on this. And it looks really clean, even cleaner than the other one, right? It's separated out these guys to be over here. So we have a nice clean tree, and they both look the same. So we feel really good about that, right? So they're doing really fast, parsimony a little bit longer, but having both of them really tells us a lot about what's going on. So finally, we want to go to our maximum likelihood tree, which is our best way of really giving it our best statistical measure of what the tree should look like. Once again, we just use the same phi-dat file. I want to make sure that everything's unique, which it should be. And then I do a neighbor joining tree, but this time I do distance equals Hammy. This is what I use for maximum likelihood. And then we use this multi D2 on the initial tree. It's just kind of working out the final. So I'm basically making a neighbor joining tree is turning it into finding the maximum likelihood. And then we unroot this. It's just more kind of setup, right? And then we basically here for our initial tree, we find any 0 or non-negative branch. We won't want any 0 branches or any negative branch links, right? So it's just kind of make sure the thing doesn't crash on us. And then we're kind of set up. This is the same no matter what you do. It's just very, very similar. And then we basically, here's our function PML, our initial tree there with our data, and we get our fit. And then we go through one more function to optimize it. And it goes through all different ways and gives us our different maximum likelihoods. And then once we have that, we basically take our tree, our fitML tree here, and then we root it, normal cells, ladderize it, make it pretty, send it up. So it's a little more complicated here, but it's the same workflow no matter when you do that yourself, right? And what do we see here? Very cool tree that puts everything pretty much exactly like we'd expect. Now, you may notice, why are there less cells? Well, that's why that unique thing we did earlier. So in a very small data set like this, with a very limited panel, you're going to have a lot of cells that have exactly the same profile, like all zeros and 1, 1 or something, right? So it basically put those together when I did this tree to make the computation simpler and the viewing simpler too, because it's kind of the same thing, right? But we got we got our normal, we got our blue, we got our green, red, everything split apart exactly as we thought, and it's much simpler here. There's our first split there, right? Where we know the MP1 comes, and there's our second split where we have the different flits and passengers to split those two. So this is a really awesome tree. I think they all look good. They all show kind of the same stuff, and I feel pretty good about believing those look really good, right? So this is the most statistically rigorous way. This would be the way you want to actually put into your cluster. And we kind of we wanted to see if anything was founding, so we looked at cells only, and we said, well, what mutation is in every single one of them? And what did we find? Our DMT3A, just like before that we talked about. And I think I saw, where did I show you guys? And we're seeing here that, we have, one truncal mutation, and then we have some specific, like clone-specific mutations that are found in each clone. And then, our number of clones is 4, 1 normal. So in our 3 malignant, we have 21 different specific ones that we can see there in general. All right. And really, just checking something really fast. All right. Okay, and so now we're a little bit farther, and lost some. I'll show you later. I just kind of wanted to show you the tree. It's exactly the same, right? You have your normal. It splits into the blue. It's just totally separate except for that DMT, and then it splits into those separate ones to go their separate ways. So you can see that there's certain mutations like FLT3-IDT here and TKD that really split up the importance of some of these, that set the foundation for this, and like things like MRAS and IDTH2 kind of set the foundation for this one. So you can go back and kind of look at the trees and see that they actually fit the labels and different things that you would expect based on. So I want you to see how you can look at the original clustering, and then the hierarchical clustering, and then finally your actual tree, and see how those all fit together. Anyway, so that really is good. And you could have different types of evolution. You could have linear mutation, where it's just a sequential acquisition of mutations. You could have neutral, where they just kind of does it randomly. You could have punctuated bursts, like you see in breast cancer, where tons of things happen early on, and then they kind of just slowly progress. Or you can have branching, which is what we've seen here in our animal data, where we have kind of a normal that's kind of just multiple subclones kind of diverge off from a common ancestor. And that's a very common way that you see this, and that's what we're seeing in AML. So now we're going to go back to COAD, the bulk things, for example, and go back to this driver versus passenger problem. So now, even with the AMLs, we had very distinct mutations, right? But we also had passengers that came along that didn't really have much to do with the actual cancer, but they might have something to do with the mutation signature, right? But most of these random events don't really contribute to cancer, but we want to find what are the true actual drivers, right? And we kind of did this last time. Let's just go into it one more time and see if we can replicate what we did there, and go a little farther as well too. So first we're going to go to our recurrence-based methods, right? How often do these things occur? Like how frequent are they? So I just basically made up a table for that. I grabbed our COAD MAF data. Remember, you could pull out that clinical mutation data right out of there. There's that function for that, but I want to just grab, all I'm doing here is just counting and dividing and making the frequencies. So I just pulled out the top 20 that had the highest frequency. And you see here, TP3, APC, KRAS, PIK3CA, the ones we'd expect. And we also see things like TTN, U6, SEN, things like that. They're just hypermutators. That's why we're seeing them up so high. So those don't matter as much, but we'll talk about filtering those out, or these others actually do matter. So frequency. If you're trying to find driver genes, they're often the most frequent. So that's one good thing to look at the beginning, right? But you got to think about hypermutators, right? So what about hotspot analysis? Well, true drivers often have hotspot mutations, right? Where the same amino acid occurs repeatedly or the same location, right? So for this, we're going to look at specific hotspot areas, right? How many times do things occur there? If things occur a lot in specific areas, they're probably has something to do with drivers. And we see here, once again, KRAS, BRAF, mixture CA, TP3, the common guys, the common criminals are up here, so that's good. So we're seeing kind of the mutations we'd expect and recurring in our hotspots, right? So, but how can we go a little farther with that, right? And look at the lollipops, right? So first we can look at the lollipops, and we see that there's just a ton, guys, right here. The R1450 is very high. That's looking really good. R1450, yes, I saw that there. And we can go to BRAF, for example. We have that BRAF 640, is a very common mutation there. And we find that things like APC mutations scatter across the gene, but cluster in these mutation cluster regions, which is really a tumor suppressor pattern where things like this V600, hotspot dominates for one amino acid in that area can really just throw things on, more oncogene behavior. But the important thing is we're seeing the same genes that you would expect in these top mutation hotspot areas here. So that's really, really good to see. We feel really good about that. But then we finally want to go to our gold standard method we showed last week, our DNDS, which is on this DNDS-CV package. And we're really trying to give an understanding of the non-synonymous to synonymous ratio, right? It really helps bring out more. This model is called variates, high mutators, things like that, to give us a better understanding of what's going on. And it gives us a ratio, basically. The higher the number, the more likely it is positive selection for drivers. So we're seeing more non-synonymous than expected by chance, right? And that's important, by chance. So to do that, we just we convert from our math object just like last time. We do our data, kind of make a simpler matrix here. We change the chromosome 1 to 1s. We want everything to be 1 or chromosome 1, right? Be the same. And then we basically ran our data there, mutations DF right there to get our result, and then just grab the 10 out of the top 10 just to kind of show you. Here's the top 10. The Q value is 0. That means it's super, super low P value. We have a high number of non-synonymous mutations, which we want to see, and we see a high number of this ratio. So the highest feel really good. Once again, P53, KRAS, APC, P53A, exactly what we would expect. TTN, we also would expect that to be high because it's hypermutated, right? But we're learning more and more about TTN as we go, right? So let's integrate this all together. So I built my own score myself, and it's basically based on all these metrics where I do things like I take the DNS score, then I add hotspots types 1.5, things like that, frequency. So I count for all these things. And we have kind of our own, you can borrow the code, our own kind of classification where I kind of take everything into account and give it an evidence score, and I sort it by that. They all have really good p-values here. You can see these have higher hotspots. So the top ones, they have high frequency, high hotspots, and good DNDS, right? And what do we see? Top 4 nailed it, right? So we feel really good about that. TTN's been pushed down. So this feels really good, really, really good. We feel like we found the right ones. So that being said, what if we want to predict cancer types? What can we predict for mutations? So now we've talked about all the ways we've looked at mutations, seen mutations, what they do. If we have mutations, can we predict different subtypes? Now I'm picking an easy example here. I'm going to pull in real pan-cancer data from TCGA mutations package and look at the mutations, the same mutations and their orientations, their frequencies in 5 different subtypes. Pull those out, and then we're going to see if we can classify based on those information. Now, it doesn't have to be this way. It could be other things. Like, let's go back to my lung cancer example. Can we classify samples that have pollution versus non-pollution? Well, they had different mutations. So if we made a model that could predict that, could we go to the next patient and predict like how much pollution they have based on how many mutations they have, or could we classify their patient based on mutations? Or can we classify even things within tumors, right? And that's just one small example, but there's a lot of ways you could have different mutations and try to classify different subtypes. One thing I also did is I used mutations to build trees, and then I basically used those trees to classify different cancer subtypes. So there's lots of models you can build mutations to make classifications. That's really what I want to get you to do. And prediction models are something that can really help knock your paper into a higher tier journal. All right, that being said, let's jump into the classification. Now, what I've done here is I basically pulled in the data from 5 different cancer types, and you won't believe this, but I've been showing you the hard way to do it, which is the most established way, and you know it's true, but this package also uses, if you pull in the TCGA mutation package that I just mentioned, TCGA mutations package, remember that was the one for dev tools, and just use the TCGA load study equals whatever. That'll automatically give you your math file instantly, right? So what they've done is they just have all the math files together in the depository for you to go grab them all. It's just a really quick way to get the mutation data, as opposed to going that long way. But be aware that the long way, they continually update that, and this may not be updated as fast. So if you're really doing a paper, I would choose the longer way. Doing analysis and learning, I think that the quick way is the best. So basically, I pulled out the math files for all those cancers, and I'm just looking at these genes. So I have 16 genes here, I believe, and I'm just looking at these only. What I'm going to do is, in each cancer, I'm going to look at these genes, and I want to know what their variant allele frequency is. And once I have all those, I want to see if I can, that's kind of the pattern of mutations for each cancer, and then I want to see if I can classify those, right? So I basically just made a mutation matrix for each of the different types of cancer here. So then we end up having a huge data set where it's just the types of mutations based on the patients in each cancer, right? And we see here's the number of samples. And we do kind of exploratory analysis really quick here. I won't go too much into this, but I'm just basically finding, I'm basically just setting up, the frequencies for each mutation, and then looking at, I'm looking at all of our genes, all of our cancers, and then just telling you, how frequent the mutations are found there. And things make sense. I think like PAD has KRAS TP3, COAD has PIK3CA, APC3, TRAS, these things make sense. BRCA has TP3, PIK3CA. So these are kind of the distributions you would expect. Based on these, can we now build a model to classify these? And that is the rub, right? So here, I'm going to try to do that using tidy models, just like last semester when I did this, and I kind of just made our machine learning data, put that together, and you can see what that looks like before we start, because we've been bringing so much together, machine learning data. See here, I have all my sample IDs, my different cancer types, and then I have all my mutations. That's all. I've kind of put that together, and I am basically splitting this into training and testing sets, right? So I'm using a 0.75 split for training data, for our training data and testing data, and then I'm going to, after that, I'm going to use that to check our model accuracy. So I am training, and I'm splitting our data into training and testing, and I am running a random forest model here, where I'm just using classification, 500 trees, and I'm going to do a tune here on these 2 values, and I set my recipe up. Basically, it's just my cancer type. So I'm going to choose the cancer type on the rest of the data. So based on all the data it has, can you tell me what the cancer type is? Because we know that is their answer. That's our result, right? And then I am basically making that into a workflow, add my model and the recipe, and I'm doing a 5-fold cross-validation. I'm going to tune my grid. Remember, all this you can find in the last semester's work. It's actually very simple and repetitive. And then, I'm going to make my grid, tune my grid, and then I'm going to choose my best parameters, which is the MGR of 4, MN of 10. So all of this was just to find my best parameters. Now I'm going to do my cross-fold validation, and I have my workflow, best parameters. I finalize my workflow, and then I run that on the trained data. I have my final fit. And then just looking at my final fit, I look back at the test data, make my predictions, and you can see here the confusion matrix is pretty good. I have about overall about 80%, right? Most of them look pretty good. Things like LUAD are off. LUAD is being misclassified as BRCA. It's probably the biggest one off. So LUAD's having the hardest problem. It might be the mutations I picked, because we're not looking at everything. We could have looked at all mutations and have a much more robust We could have, we could put 1,000 mutations in there. It would have been better, right? So anyway, 80% is still pretty good at getting those. And you can see kind of the number correct. Accuracy is the best in BRCA, worse than STM. I mean, worse than LUAD. But anyway, and here's some of the features, that are just important for some of the predictions for the different cancers. It'd be better to probably not look at these per cancer, but anyway, you take a look at those. Here's the ROC curve. It's just kind of showing you, obviously the LUAD is the worst. The higher the better. You're seeing some of the better ones like BRCA. So it's working pretty good. Not the best ever, but like based on barely tuning the model at all and just looking at 20 genes, 16 genes, it's pretty good. And really, it's just showing you what's possible. That's a really good prediction, but there's better ways to do it, and you could do better than that. And let's see here. Let's move forward. So now what I want to do is talk a little bit about, we're going to practice problems, just kind of dive in a little bit more. The first thing I want to do is look at APOBEC signatures, right? Now, because we kind of talked about them before, these are enzymes that normally function in innate immunity, and when dysregulated, they cause mutations with a characteristic signature that ends up being the SBS2 and 13, right? Now, colorectal cancer has a very low APOBEC. That's why I made a point of saying that it looked weird when we saw the hypermutators. Now, bladder cancer is different. It has a very high APOBEC, so we're expecting to see the higher number of these signatures mutations in this cancer. So let's take a look at it. First, we'll load up our BLCLA. All I did was change the BLCLA and nothing else. Got our math file, just like COAD. We're cruising along just like before. Then I'm going to do my trinucleotide matrix with my thing before, just like before, no difference. This time I have 414 samples times the 96. Then I'm going to fit this just kind of a different way. I'm going to get known signatures. And then I'm going to fit the signatures, right? So this is just a different way you could do it as above. I just want to show you the different options, right? And then we're basically going to convert, because we want percentages here. Now we have our signatures now. We're basically just going to look at the contributions of each signature and change that into a percentage. So it's very similar to before. I used a couple different packages. They're all in math tools, and it's really almost exactly the same thing. I just want to show you the options. And this is just a bunch of data wrangling. The idea is we went in and we fixed signatures, and then we looked at how many samples. We looked at every sample, and we saw like the percentage of SB2, SB13, and then add that up to the total, right? And we sorted by the total APOBEC. So these have the highest APOBEC changes, and then we see the difference between the 2 and 13, the different ones, right? And then I want to classify these samples as high or low, and I'm basically going to set a really low threshold of 5%. So if they have 5% percentage in there, they are going to make it into the high. So that puts a lot in there, puts 93% in there. 65 is the normal value for it. So if you count for those hypermutators, that's probably what's inflating that value, or my 0.05 could be a little high. I could bring in some of the more low ones there. But we're just kind of want to see a distribution of them. Look at the distribution of all of them. So now, if you see now, the APEC contribution is pretty much the same across everything, right? There's no high values in just one spot, and that's good. A broad distribution with many samples, it confirms that this is a high APEC cancer type. So we feel good about seeing that, right? And if we compare the high and the low a little bit more, I'm just going to look at the actual signatures themselves, and you see the difference. You see the difference here, this guy here, the guys here. That's really setting up your high and your low APEC. So your high APOBEC are bringing in some of these new signals here, and then the low ones have some different signals. But you can kind of see the trinucleotide difference, and you can kind of see how well they're correlated. And one thing we're seeing here is that CAG and CT transversions or whatever transitions, we see much higher changes here. Where the CA, we see much higher in the low, which makes sense because these are the two we were looking for, especially if they're in the right sites at TPC. Okay, so let's just kind of listen to the understanding of the APOBEC stuff. Let's kind of we're going to find the TMB, basically just like before, and do the correlation between higher TMB, higher APOBEC. And we see it's significant and has a row of 0.566, which is there, but not super strong, but it is there. So as you see, as the tumor mutations go higher, the APOVEC tend to go up a little bit. It's kind of slow, but they do go up. And you see this one super high mutator up here with low APOVEC, but super high TMB. But anyway, there is a mild correlation between these two things. If you actually look at the medians of the TMB between high and low, we see there is a significantly higher in here. So we can say that the TMB really is associated with the APOVEC high status. But remember, part of that is because of the high mutators. You really need to probably clarify that or account for that as well too. So let's jump back in and redo that plot APOVEC difference with our bladder cancer. And now we're seeing much more logical values. 53, 161. It's still inflated because of the TTN, but it's more logical what it could be. So you can either take TTN out and redo the analysis or account for, you can actually model for mutation number as well too. The different things you could do. But like I said, bladder cancer is already high, so we got values that made sense there. Reflector is actually low, so our values were misrepresented by the obvious hypermutators. So another thing I want to look at is just the different ways and approaches to that phylogenetic analysis with AML data. Remember, we use the Jacquard data, Jacquard distance, same as binary, which is really great for binary matrices. So I wanted to try it also Euclidean, which is normally good for continuous data. I like using it for things like, it's good for mutation data that has like Villa frequencies, things like that. Manhattan's really good for outliers. And so that's really good there's a lot of outliers in copy number. So I think Euclidean does pretty good for the most part for RNA, I think. Jacquard, good for DNA, and Manhattan, good for copy number. That's super general, but there's logical reasons behind it. So first thing I'm going to do is make our distances, right? So we're using our matrix like before, and we're just choosing 3 different methods, and then we're just making our trees. We're basically just making our trees with 3 different metrics. Just think of it that way. And so I got my trees, and I really get my distances. I'm making my trees. I'm basically throwing this all into one thing. I'm doing neighbor joining, Jacquard distance, and then I'm also rooting it with a normal cell. And doing that with 3 different ones and plotting them, and now you see the 3 different ones, and they look very similar. I would say that if you could see, the Jacquard does a better job of handling, the normals that are out, and it kind of cleans up some of this stuff. You notice how messy the green is? It kind of cleaned it up. So really, it's finding ways to minimize the branches a little bit better and cleaning up data a little bit better. You can kind of see why it's the best. There's not a huge difference in the error, but there is a difference that you can see, and the more cells you have, the more you will notice. One last thing you can use is UPGMA. It assumes a molecular clock, so that means that everything evolves at the same rate, which is impossible and not true, but this is the most simplistic way to look at this, and a good way to know how it works too is it's a agglomerative clustering, which means it goes from top down or bottom up, and here it's top down, where just every single split, it chooses the most logical split. The idea is this is a heat map. So if you turn this computer on its side imagine this being the top of a heat map, right? Or even the left side of a heat map. This is exactly how things like feat map and complex heat map if you choose like average distance, this is exactly the kind of thing they're giving you. So what's cool about this is you can match your trees. If you want to match your tree perfectly to your heat map, you can use UPGMA, and you can have a nice tree that matches your clustering, your matrix, everything. So that's just an idea of why you want to do that, and it's just nice to see it all coming clear. There's one little mistake. There's a little gray mistake there if I look really in. So it's not perfect, but it's looking pretty good, right? So that's just another way you can do a simple expression. Finally, one of the most rigorous ways you can do this is bootstrap. So if you have a really great paper and you want to really prove a tree is real, the way you would do this is first you need to unroot your tree, your final tree, and then you run it through this boot phyllo. And there's kind of a function here where I'm just doing the same thing. I have an unrooted tree, right? Unrooted neighbor joining tree, my normal matrix, Jacquard distance, and I'm basically running this 100 times. And And then I'm basically looking at the similarities between every single branch change over 100 times. You can do 1,000 or whatever. So the idea is if things change, if things stay more the same over all the trees, they should have a higher confidence that they're real. Like those splits feel real. So sometimes early splits have really high confidence because you have more belief that they're real, right? So that's an example of that. And we did this in our tree, and we did have our 3 clusters look really good. But if you notice, our bootstrap values aren't super great where we would like them. We like the ones early on the splits to be really good. But this is a great teaching moment because bootstrap doesn't work good for small panels. So you need a lot of genes. So that's the way these methods really worked with when they had different taxa and tons of different samples. So remember, our mission bio set has 200 cells, but only 30 columns, only 30 amplicons, right? So what's happening is, I forgot to tell you that each time we're doing this, we are bootstrapping, we are sampling from the tree. We're not taking everything. That's a really important thing to tell you. So we're basically sampling out some of the columns, making a tree. And then we're putting them back in, and we're sampling with replacement, again, 100 times in it. So the idea is if you just change up the features a little bit, the tree shouldn't totally change, right? And the fact that ours even stay the same at all is really good. But you can imagine if you had 1,000 columns, 1,000 genes, and you kept sampling the replacement, there wouldn't be as much change, right? So you have more belief, there'd be more higher bootstrap values on more of the clear changes. So that's a good example of what you expect and why we don't see that in our data, here. And then finally, let's identify clone-defining mutations. We kind of mentioned earlier there's mutations that went with certain ones. We looked at DMT3. We just looked and said, hey, what's found in every single one? Well, DMT3A is found in all 3 clones, so we know that is the one that matters. And then we also looked at clone A, B, and C, and we found what are the unique mutations in each one of those clones and kind of plotted them out here. So we kind of got rid of our passengers, and these are the ones that actually mattered to split up our guys. And you can see here, normal is moving along fine, where this green clone has, DMT3A, MP1, FLT3, TKD. The blue, the green have all of these, which kind of makes sense. So if you remember, the blue split off early, right? They all have DMT3, but blue split off early and has its own mutations by itself. Green retained the same mutations here, right? But then split off, and one did FLT3 tyrosine kinase duplication, and one did intertandem duplication. So the FLT3 did something different in each one of these, and the implication of this is the treatment. It's going to be different. So there might be different treatments for different clones, and if you might hurt one clone, might help another. So it's important to know the distribution of these clones and what is going on and what's really driving the tumor, right? So finally, let's do our last problem. So based on that cancer classifier, how can we do better for it? How can we do better with that classifier? And this is just more teaching you how to do prediction. So how can we add more value to that prediction? Well, add more features, right? The more features we have, the more data we have, there's a good chance we can do a better prediction. So I'm going to add TMB to each one of the cancer types. They're going to have different TMBs, right? So I'm going to add that sample level and also add pathway features. So each cancer type has different pathway features, different mutations associated with those pathways. So we're going to add those in, the different mutations, and we're going to see if adding this information can help. I basically just made a function to calculate the tumor mutation burden, which is basically just dividing mutation count by 38. Put that into a new tibble with engineered features as well. So you see the different pathways here. So now we have those associated with the different cancers as well. And I'll let you guys kind of go on a little bit later, and I won't just keep it forever talking about this, but I basically just put everything into one matrix, split it, did the same things before exactly. It's really the same code, really. And all I did this time was did my prediction based on the training data set for the additional values here. And this time I got 82%, pretty much the same, a little bit higher. And then I wanted to say, okay, well, what if I compare that to XGBoost and SVM? So I basically made XGBoost, SVM, and the same thing, same splits, same recipes, and just ran the same thing through each one of them, got the predictions, and here are my values. Random forest is the best. They're pretty much all the same. And, accuracy is, I think accuracy is not the best way to look at this, but I think it's for teaching purposes, it's just fine. And one thing I did, a nested thing. What does nested mean? So nested basically, it's very subtle, right? So it's kind of complicated here. Let's try the best way to explain this. Standard cross-validation has a kind of a bias because you use the same folds to tune the hyperparameters and estimate the peak, right? Either you split up your test data, have a validation data set, and do a better job at that, or you can nest it. And we're going to show a way you can nest it where you can have the inner loop can be 3 folds. It can tune the hyperparameters, and the outer fold can be the thing that once the model's tuned, it can evaluate on truly held out data, which is really just like a validation set, right? It's similar. And the nested accuracy is typically lower than your standard cross-validation, but it's more honest, right? So nesting, you can do nesting, or you can add, train, test, validation data sets. So that's kind of an equivalent to nesting. It's pretty much the same thing. So let's go ahead and do that and just see what we get as far as the result for that. So let's see, I ran through that. And really nesting that, I didn't give it, let's see where I did it. My SDSR, I went below it, right? So here's my result, here's my outer folds. Then I set my tunable trees, grid. I mean, this is all just using, let's see, it should be, so what did I do? I guess I use XGBoost here. Yeah, for this one, I used XGBoost. I'm getting confused. That's the parameter. I ran it, I did my outer train, outer test, and then I final thing on my infolds. I tuned my XGBoost, and doing all of that parameter, I get a final accuracy of 0.81, pretty much the same as before, but at least we have more belief in it. I think that's the important thing. And we look at our matrix, we're kind of seeing the same thing, seeing the same issues. So TMB, those things help, but they didn't help that much. They didn't help as much as we'd like because we're still seeing these problems. But if you look at it, it's actually better than I think the misclassifications are looking a lot better. Still have the 22 misses up there. For Luad BRCA, the 22 misses. Yeah, Luad BRCA miss. So we still have kind of the similar values, but it's a little bit better, but at least it's more believable, right? And here's just kind of you can look at the frequency profiles by cancer type if you want to look at those, get a feel what they are. And kind of talks about some more classification areas, why they happened, why they didn't happen. This is a good learning experience to understand why the predictions worked. In real life, we probably use all the mutations for those and not just a few. But I just wanted to give you a small example, so you want to do that. But this is a really good learning experience, I think, to really look at that and see what is going on there and what are causing the problems. So they're usually pretty obvious. And common pitfalls, use the right distance, root the trees, look for your problems and leakage in your cross-validation, and really map it back to biological context. Here's some kind of key publications you can look at, some software documentation that might help you a little bit. For instance, like here's the GitHub for the mutations. Fanghorn is all your phylogenetics. Tidy models for those models I built, the prediction models. Mutation patterns, sig minors, working with mutations. And these are places you can find a lot of that data here. And I think that's about it. This is the RNA workshop series completed. We've gone through pretty much everything I would do with mutations. Like if someone gives me a mutation set, what do I do with it? Once I get it all normalized and I feel good about it, then what do? Well, I make an oncoplot. I look at correlation analysis, make a lollipop analysis. We look for things like hypermutators, we do mutation signatures, and do clustering, dimension reduction, visualization. We do, what else can you do? Survival analysis, pathway analysis. Phylogenetic tree, right? Okay, good. That's pretty much all you can do. Those are all the things you really can do with mutation data. And you can do with bulk or single cells, not that different. The methods aren't all that different. It's not as big of a jump as it is from single cell to bulk RNA. That's why I didn't split it up completely. It really overlaps a lot. So yeah, by what you've learned, you really should understand these mutation data fundamentals. Things like the MAF files are so easy to work with, but you could also use any type of format, and I can help you orient that to what works best for you and what you're trying to do. Learn the statistical, patterns, clinical associations. Those are kind of various things that you would find to get a good feel for what's going on in the data, and then finally looking at look more into mutation patterns, the trees, and really put this in a state of like mutational processes and really tumor evolution. Tumor evolution is the best way, or one of the best ways, to understand cancer progression and how to fight it, I believe. And I guess that's it. This is going to be our DNA workshop, and we will move on to do things like copy number workshop and RNA workshop next. Thanks everybody for attending. I appreciate it. And I guess that is it for today. Thank you.